Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today, I am going to talk about the types of lipid part 2 from the paper Lipid Metabolism. The objectives of today's talk is to know about the complex lipids. Actually, if we look at the type of the lipids, they are categorized into three different types, and these are simple, complex and derived. What are the significance of the complex lipid and how they act in a system is the part of discussion today. Complex lipids, they are further categorized into different types like phospholipids, glycolipids and other lipids. So one by one we will discuss the type of complex lipids in this module and in couple of the forthcoming modules. In the last module, we have discussed about the simple lipids and a brief introduction about phospholipids. Today, we are going to discuss about the glycolipids. Actually, glycolipids are the lipids that contains carbohydrates or the saccharides. The carbohydrates that are present may be either galactose or glucose. So in a sequential manner, we will discuss today the different types of glycolipids. So we are discussing different types of lipids, and these types include the simple lipids, complex lipid and derived lipids. The simple lipids, including the fats, the oils and the waxes, have already been explained in the previous modules. In the complex lipids, which includes the phospholipids, glycolipids and proteolipids, has been discussed herein. Phospholipids are phosphorus containing lipids, whereas glycolipids are lipids containing saccharides or the carbohydrates. When lipids are amalgamated with proteins, it forms a complex mixture of lipids which is called as proteolipids. So in this module we are discussing about different types of glycolipids, and it has been continued from the previous modules. To continue the glycolipids, as I have told before that a glucose or galactose moieties are present in this kind, and a diglucosyl diacylglycerol estulides has been reported from the kernels of the oat. This diacylglycerol estulides includes a linoleic acid cluster and a 15 hydroxylated linoleic acid, which is also called as avenololic acid. This esterolide is abundant in the kernels of the oat. The structure is complex. As you can see here in this slide, that 4 parallel chains are running, which is linked to a carbohydrate or a saccharide moiety. Among the complex lipids, apart from diglycerol from the kernels of the oat, 2-acetoxy, 1,3-glycosyl oxo-octadecanoyl glycerol has been isolated from the exudates of the secretory organs or the granular trichromes of Cerasu yedoensis, which is a cherry tree plant found in Japan. The disaccharide moiety of this 2-acetoxy 1,3-glycosyl oxo-octadecanoyl glycerol is linked to a 3-hydroxy octadecanoic acid, which esterifies glycerol in SN1 position. However, the position SN2 being acylated, as it has been seen in the structures shown below. Among the complex lipid, especially the glycolipid, there is another subtype which is called as alkyl acyl glycosylglycerols, in which cramerides, which is an intermediate form of monoglycosyl diacylglycerol, was isolated from a sponge named Pseudoceratina crassa, and this crameride is having an unusual cyclotol moiety, a branched alkyl chain and a saturated acyl chain with either 14 or 16 carbon atoms. And these carbon atoms may either be branched or unbranched. These cramerides compounds also act as a feeding deterrents in fish. Dialkyl glycosyl glycerol are the diether lipids which has been described from bacteria. They contain the glycosidic head groups noticed in carbonated chimneys of the hydrothermal field. These compounds have non-isoprenoid diethers and hence signify an exceptional permutation of archaeal distinctiveness in the lipids of bacteria. A mono or a di glycosyl moiety is present at the polar head group and alkyl chains, which are 14 to 18 carbon units. And these alkyl chains is either monomethylated, monounsaturated, or saturated. Since these glycolipids exist from the creation of life on Earth, it was contemplated that they were the evolutionary precursor of phospholipids, and their existence is interrelated to an incredibly squat level of phosphate in the atmosphere. To continue with the glycolipids, another type, or rather subtype, is alkyl diglycosyl glycerols. In this subtype, myrmecocytes, which is a novel glyceroglycolipid, have been reported from a sponge named Myrmecioderma species. This myrmeciocytes possess anti-tumor properties. An alkyl ether chain in which 16 carbon moiety with or without a branched methyl group is present at carbon number 1 position of the glycerol, whereas 2 moles of glucose at carbon number 2 and 1 mole of xylose at carbon number 3 is present. As has been very clearly seen in the shown structure. Another class of the glycolipids are the glycerophosphoplazmalocytosine. This trikentrion loaf is a Senegalese sponge, and from that sponge an exceptional ether lipid has been revealed, which is distinguished by the glycosylation of 2 glycerol radicals and an alkenyl chain of ether, which consists of 24 carbon units with single double bond present at the third radical. In human brain, glyceroplasmalocytosine, a plasmal conjugate of cytosine and glycerol, which is an additional ether lipid, has been depicted. Cyanobacterium, Cyanocystis species possess acylated forms of glycoglycerolipids bearing a palmitoyl group which is esterified at the hydroxyl group of carbon 6 of the terminal glycosyl moiety of either acylated DGMG or acylated diglycerol moiety. The presence of acylated MGMG or MGDG and DGDG was also stated from the nitrogen fixing cyanobacteria and the homogenates of the leaves. Bacillus acidocaldarius also possess an extraordinary glucosamidal glycolipids, which comprise of 64% of the total lipids, which are N-acyl derivative of glucopyranosyl glucosamine diacylglycerol, along with the amide-linked branched 13 cyclohexyl tridecanoic acid, 11 cyclohexyl undecanoic acid or heptadecanoic acid. In algae as well as in bacteria, a huge amount of glycolipids, including diverse combination of sugar molecules, have also been reported. Another important type of complex lipid, or rather say a complex glycolipid, is mycoloyl arabinosyl glycerols. These mycoloyl arabinosyl glycerols clearly indicates its association with that of mycolic acid. These mycoloyl arabinosyl glycerols are the new glycolipids which has been isolated from the complex of Mycobacterium avium and Mycobacterium intracellulare. The characteristic feature of this glycolipid is described by an arabinopyronosyl glycerol, which is acylated on the 5 carbon of the arabinose moiety by an assorted form of mycolic acid. And these mycolic acid may be the ketomycolic acid, simple mycolic acid, or maybe the ester form of mycolic acid. As you can see, the structure represents the unique triangular alternate carbon association along with the presence of a sugar moiety. Another important class of glycolipids are alkyl galactosyl glycerols, that too with the presence of acetyl group. The example of this class includes plasmalo-galactosyl alkyl glycerol, which is a novel galactosyl alkyl glycerol tailored with a long chain cyclic acetal at the sugar moiety. This plasmalo-galactosyl alkyl glycerol was isolated from the brain of equine. This subtype of the glycolipid is having an acetyl and alkyl group, the chain length of which were 14 carbon for the alkyl group and either 16 or 18 for the acetyl group. The intact equine brain consists about the 5 milligram of plasmalo-galactosyl alkyl glycerol. The example structure is shown below. Phosphatidyl monoglucosyl diacyl glycerol, which is abbreviately known as PMDG, is also a subtype of glycolipid, which is a part of the complex lipid present in many gram-positive bacteria. However, the structure and the metabolism of phosphatidyl monoglucosyl diacyl glycerol were elucidated in a gram-negative bacteria, Pseudomonas diminuta. The structure of which has been shown below. Another important phosphatidyl monoglucosyl diacyl glycerol is phosphatidyl glucosyl aminyl glycerol, which is also abbreviately known as PGG, and it has been found in Bacillus megaterium species. N-acetyl glucosamanyl phosphoethanolamine diacyl glycerol is another subtype of the glycolipids, which is present in the Clostridium tetani species. Clostridium tetani is the causative agent of tetanus. and from this bacterium, a derivative of N-acetyl glucosamine diacylglycerol has been isolated, in which a phosphoethanolamine unit is attached to the 6th prime position of the sugar, which can be seen in the shown structure. From the several species of Halomonas, which is an halophilic gram-negative bacteria, phosphatidylglycerol moiety has been isolated. It consists of a phosphatidic acid attached to the glycerol residue that is alkylated by a glucose moiety, and oleic acid and palmitic acid are the most plentiful acyl chains attached to the phosphatidylglycerol moiety. The tetanus causing microbe Clostridium tetani produces N-acetyl glucosamine diacylglycerol, where a phosphoethanolamine moiety is linked to the 6th carbon position of the sugar. In phosphatidyl inositol polymannoside, which is also abbreviatedly known as PIPM, has been established in Mycobacteria. At 2' position of inositol, a mannopyronoside cluster, and at 6' position 1 to 5 mannoside groups are present. It was established that phosphatidylinositol mannosides from Mycobacterium tuberculosis wield an effective anti-inflammatory effects and inhibits the production of chemokines, cytokines, and nitric oxide, both in vitro as well as in vivo, via toll-like receptor inhibition. Mannophosphoinositides presence in the phospholipid composition is a striking feature of some bacteria and actinomycetes. Monomannosides have been delineated in the Streptomyces, Propionibacterium, and Mycobacterium species. Two types of dimannosides were identified from Streptomyces griseus, with one extra fatty acid fastened to a mannose residue containing either tri or tetra-acyl mannosides. In Mycobacteria, polymannosides, which are one of the most complexes, with a number of fatty acids. Acylating the mannose chain were identified. In Corynebacterium urolithicum, acylated phosphatidylinositol dimanosides were also illustrated with either 16 carbon or 18 carbon atoms, acylating one of the two mannose group. So, today we have discussed and learned about the second part of the types of lipids. In this part, we have explained how the glycolipids, which is a part of complex lipids, play an important role. The complexity of the glycolipids and its presence in different organisms reveals that numerous different types of glycolipids occur in nature, and that what makes and gives the complex lipids its name. This type will be continued in a couple of forthcoming modules, and we will learn how the lipids and its different types occur in nature. Thank you.